Phytophthora infestan, the pathogen of the lead blight disease and its life cycle. The domain of Phytophthora infestan is Eukaryota, kingdom Chromoviolata, phylum Heteroconthophyta, class Oomycota, order Peronosporales, family Phytaceae, genus Phytophthora, and its species name is Phytophthora infestan, which is the description of this pathogen is the branched spongiospore which failing at the point where sporangia were attached are distinctive for Phytophthora infestan and useful for identification of this pathogen. This pathogen causative agent for plant disease which is leaf blight. About its habitat, it can grow wherever potatoes are grown, thrive in cool weather and wet conditions. What about its nutrition? It obtains plant nutrient by using hyphae. This pathogen does not obtain very many nutrients from the host until it establishes itself within the epidermal cell of the host. When it has established itself, hyphae can readily uptake the nutrient. Required nutrient by this pathogen is organic carbon, oxygen, hydrogen, phosphorus, nitrogen, sulfur, and so on. Okay, let's talk about reproduction. This pathogen grows in plant tissue to grow in. It's usually reproduced asexually. The life cycle usually begins as the sporangia are carried by the wind, land on the plant tissue, and released by zoospore, which is indirect germination. It occurs when conditions are wet and cool. The spores are biflagellate, having both tinsel and witch-flesh flagellum. This is the characteristic of Oomycota. The spore will swim until they encase in the host. Reproduction of the pathogen involves asexual and sexual. In warm condition, Phytophthora infestan will infect the plant by direct germination. The asexual life cycle of the Phytophthora infestan is characterized by alternating phase by hyphal growth, sporulation, sporangia germination, which is either zoospore release or direct germination, example germ tube emergence from the sporangium, and the re-establishment of hyphal growth. Second is sexual reproduction. Sexual reproduction occurs when both mating types are present. The antherium and oogonym, the only haploid part in the life cycle of Phytophthora infestan, which is nuclei fused together. It means the process of karyogammy. When the antherium enter the oogonym, then they form a diploid oospore which develop into a sporangium, and the cycle continues as it would asexually. So the asexual part is the karyogammy part.
This is potato leaf blight disease cycle. So zoospores are produced and released from sporangia, and then they infected leaf and also tubers. From that infected, the seedlings produced by infected tubers become disease. Also leaf have lesion and fungus develop on it. So from that, sporangia will disperse to another healthy leaf. So this is the cycle of potato leaf blight.
Field symptoms. There are 7 field symptoms in this slide. The first one is light to dark green, circular to irregular shaped water-soaked spots. The third, leaf lesions frequently are surrounded by a halo chlorotic halo. Lead blight lesions may be present on petioles and stems of the plant, especially in new growth where moisture persists. During active lead blight growth, especially in cool, wet weather, a white mildew appearing area is visible at the edge of the lesions. 6. Lead blight infections of tubers is characterized by irregularly shaped, slightly depressed areas that can vary considerably from brown to purplish of variable size on the skin. Lead blight causes a tan to reddish brown rot found under the skin. So the next slide is showing the picture of an example of the initial symptoms of lead blight are small, light to dark green, circular to irregular shaped water-soaked spots. This is the picture. The next is the leaf lesions frequently are surrounded by a yellow chlorotic halo. The third picture shows that lead blight lesions may be present on petioles and stems of the plant, especially in new growth where moisture persists. Next, during active lead blight growth, especially in cool, wet weather, a white mildew appearing area is visible at the edge of the lesions. Lead blight infections of tubers is characterized by irregularly shaped, slightly depressed areas that can vary considerably from brown to purplish of variable size on the skin. As in the picture, you can see that lead blight causes a tan to reddish brown rot found under the skin.
Conventionally, detecting disease on crop leaf are based on visual assessment and diagnostic method. Diagnostic testing methods include polymerase chain reaction (PCR), enzyme-linked immune sorbent assay (ELISA), fluorescent in situ hybridization, and biomarker-based detection technology. However, these methods have shown to be time-consuming, inefficient, and destructive. Also, highly trained and qualified technicians are required for such detection technique. Therefore, an advanced method is needed. Advanced method that we use is called hyperspectral imaging. As you can see at the picture above, hyperspectral imaging (HSI) is a technique that analyzes a wide spectrum of light instead of just assigning primary color, red, green, blue, to each pixel. The light striking each pixel is broken down into many different spectral bands in order to provide more information on what is imaged. But there are some diagnostic tests that be developed by commercial company to each detect plant disease. It was called Phytophthora Rapid Test, which is genus-specific test designed for the in-field detection of many Phytophthora species. Not only for Phytophthora infestan, lead blight, it also includes Phytophthora ramarum, southern oak dead, Phytophthora kernovia, Phytophthora lateralis, Phytophthora cinnamomum, Phytophthora cactorum, and others. It can detect Phytophthora in a wide range of plant materials including woody tissue, leaf, and roots. Using the test is simple. As you have to do is break up some plant materials, then add the sample to the buffer bottles, then shake the buffer bottles include ball bearing. Then you need to allow the plant material to settle in the bottles. Finally, use the pipette provided to add liquid to the test. Then you will get the result from this test.
Management option available: plant lead blight, free seed tuber, disease resistant. Do not mix seeds lots because cutting can transmit lead blight. Use a seed piece fungicide treatment label for control of lead blight. Eliminate source of inoculum such as hairy nightshade, wheat species, and volunteer potatoes. Management option: targeting disease cycle. Fungicide. The fungicide have to be put on early. Application made before row closure result in better coverage and are more effective than application made when an extensive canopy is present. Protectant need to be applied before the lead blight spore are on the leaf surface and germinate. When spore is already present, the protectant will trap it on the leaf surface, and the germinate spore will enter the leaf, eating it out. The protectant are great, but if lead blight spores are present, we have to use the systemic fungicide. Vine desiccation. Dead vines are essential since the lead black fungus need living tissue. Spore can fall off the vine to the ground and infect tube while still in the ground. Tuber can be infected during harvest by contacting infected living vines. Dicot is recommended and the addition of copper sulfate will aid in the killing spores. Flaming and sulfuric acid will kill the vine faster and will reduce exposed spore only. Not a faster vine kill will not cause the tuber from the mature faster, such as shed skin. When desiccating of potato field, kill any nightshade even along the perimeter road and fence. Cool piles. The risk of lead blight can be greatly reduced by eliminating source of inoculum. Presently, the lead blight fungus is only known to overwinter within infected living plant tissue. In potato production areas, infected tuber from the previous growing season are the most important overwintering hosts. Therefore, elimination of both cool piles and or any other source of volunteer potatoes is one of the most important methods used for lead blight control.